Cool. All right. Cool. Thank you, guys. Good morning. So this is our week at a glance for week 4. I'm not going to go into it in too much depth. We have 2 different main labs that you guys are going to be doing. In biology, we're going to be doing some concept mapping, assisted with your TAs, in order to help you guys start preparing for your biology exam. We'll talk about it more as the time comes, but this is really one of the methods that I think you guys should be using to help prepare for these exams. We'll help demo it for you. It's also a great instructional technique because it allows us to sort of visualize how you guys are thinking about some of the class material, and it's a really effective way for us to diagnose any misconceptions that might cause you to later lose points on an exam, this type of stuff. But so that's what we're doing in biology. In chemistry, this is the first of our 2 main interdisciplinary labs for the semester. We're doing the first part of the plant pigments lab. We will do the second part of this the following week in biology. This is a good lab to use the techniques that you guys have been working on for the first couple weeks of this semester. Approach it professionally because you'll need data for this for your final poster. If you don't get data, I'm sure there are other things we can do to make sure that you guys don't fall behind. But do respect this lab, come prepared, and we're dedicating today's studio in order to help prepare for that. Also, as a reminder, you guys have your chem exam on Monday, right? So you guys have been probably started studying for that. What types of things have you guys been doing in order to study? Who's been reading the book? How about doing extra homework problems or reviewing homework problems? Okay, a few less. This is one of the big things that I want to promote to you guys. Studying for chemistry is it's going to be application level skills that are frequently taught. There's going to be very few questions that are saying something along the lines of what is an orbital or what is a molecule. The vast majority of the things on that test are going to test your ability to use things. And if I know Dr. Daly, he's been modeling the types of things that you'll need to do on the exam in class. So those in-class problems are the types of things that you're expected to do. That said, in class, he generally asks you to do those as you're learning them. It's not like you'll be learning new material on the test for his sections, but you do want to go back and review the types of things he's asking you to do on those class worksheets. That's probably a good way to start studying. Cool. So, if we keep moving along here, and this doesn't want to control my computer for some reason. There we go. I'm going to make sure that both that week at a glance and this slide make it up onto the front page of your Canvas site, or onto the modules page of your Canvas site. I think you guys have a good idea of what to do in chemistry. Just as a reminder, we do have both sets of assignments for week 2 and week 3 due for biology because we had pushed them back the week before. All of those assignments and quizzes should have been set so that even if you had submitted them early, you can resubmit them. Remember, for the group figures, we just need 1 submission per group. The last submission that you guys submit before the deadline is the one that will be graded. All right, so what we're going to be working on today is the pre-lab for your pigments assignment, and you're going to be, your pigments lab, you're going to hear a little bit more lecturing from me than I'm used to because I think this could help you supplement your understanding for this. So definitely notes would be a good thing to work on. You could either take them, so either a notebook or honestly just working within this lab, you can probably take notes here. This is on page 44, and you'll also want to take your eye clickers out. Cool. All right, guys, let's focus attention back up here. So who recognizes these molecules anyways? What's the one on the left? Your left. Acetone. What's this guy over here? Hexane. You'll do more organic naming, that type of stuff in organic chemistry, but so we're probably not going to test you on your knowledge of this kind of stuff now, but hopefully these are familiar to you if you started reading through this pre-lab assignment. So we're going to ask kind of a simple question here. Of these, Which of these is more polar? Yeah.
Acetone.
So acetone, why?
It has a oxygen-carbon bond.
Okay, has an oxygen-carbon bond. What is it about those oxygen-carbon bonds that make them more polar than others, or than many others? Yeah, so there's a difference in electronegativity. So I'm going to pull this up here, just quickly. So there is this idea when you're taking a look at one of these electronegative bonds that if we have a carbon and an oxygen here, there's going to be a relatively large difference in electronegativity such that the oxygen here is capable of pulling more of the electrons closer to it, creating a dipole. Oh, that is not working on here. Sorry guys, this was supposed to be projecting. So, move over here to the board. The idea being that if you had an acetone, something along these lines, this oxygen is much more electronegative than that carbon, meaning that it's going to pull more of the electrons closer to itself, leaving an asymmetrical distribution of those electrons. That kind of leaves this thing partially negatively charged on the oxygen, partially positively charged on the carbon. This gets to be really important when we start thinking about solutions because this now might be able to interact with something, especially polar solvents like water. Right? So we have a similar polar bond here, right, where this oxygen is pulling more of the electrons towards itself, meaning that this partially negative portion of the oxygen would preferentially associate itself or find itself near other partially positive sites. That's kind of the fundamentals behind polarity. It's one of the real things that you're going to want to get and take away out of this semester. It definitely is going to be one of the big things that explains this lab and a lot of the next probably 3 weeks or so of what you're doing in chemistry. So what I'm going to ask you guys to do is try and think through some of these questions. We'll do some simulations, but this is emulating about what we're doing in lab. We have this big chunk of cellulose. This is paper. That's one of the things you should kind of pick up out of this. But we have this big thing of paper, and we're about to dip it into a liquid. What do you guys think is going to happen? All right. All right, thank you guys. So you guys said, by and large, that the water's going to travel up the cellulose. Good. Now, we can try and run this. It should work. There we go. So get used to seeing animations like this. I've got a couple of them over the next couple of slides. This is an easy one, right? We should know that if you dip paper in water, that water is going to wick up that. You'll get a little bit more into why that happens in the next couple of weeks in chemistry. For right now, knowledge of that is something that's going to happen is sufficient for where we're going today. So if we move on here, what happens if what you had down here wasn't just a liquid, but a liquid with some type of a solute in it? What do you think might happen? There we go. All right. Close enough to everybody. All right. So the split on this question probably comes from the fact that it does depend a little bit on what the solvent is and what the solute is. If we were thinking just a general one, there's probably going to be some force exerted by our solution on the solvent, on the solute as it moves up the paper. I would think about this as kind of the general bulk force that's occurring within the lab that you guys are going to be doing next week. If there is something dissolved in that solution, unless it comes out of solution, it's going to be pushed up the paper as your water or whatever liquid you're using is wicking up that paper. Again, I don't necessarily need you guys to have known that coming in, so don't feel bad if you're getting any of these wrong. We're just kind of trying to demo and help you guys make observations with us. So this is a really quick video of a little bit of what's happening in the lab. You guys are going to set something up very similar to this, where you have a tube or a small jar with some solution at the bottom. You'll have ground some pigments onto that paper, and you see the level of the water, or the level of the solvents in here, wicking up the paper. What else do you guys notice about the pigments here? Yeah, the pigments are moving as well. And the goal here, this is a really nicely set up one, our goal is going to be to use this as a means of separating out some of the different pigments. So what we're going to go through in the next series of slides is a little bit of, let's stop that before it autoplays, is we're going to try and scaffold and figure out how we can maybe make this happen. Right. So let's keep going. So we're also going to start throwing some terminology out here. So when you were taking a look at that, that's paper chromatography, is that technique, you have two different phases of this item, basically. You have what we call the stationary phase. That's going to be the paper. And you have a mobile phase, the things that are moving up it. I'm going to try and use both pairs of terminology here so that you stay on the same page. By the time you get into lab, try and be comfortable with the idea of calling these things the stationary and mobile phase. It'll help your understanding out. So, what's the polarity of this thing? There we go. All right. It's okay that not every single person responded. We've got a bit of a range of responses, and this is a good thing because there's not a simple answer to this question. So, who thought this was polar? What are some of the things that you found on here that were polar functional groups? Yeah, there's lots of these OH bonds, right? We just talked about these. This is a highly polar bond. There's some others in here as well, but there's definitely polar bonds. What about the groups who thought this was nonpolar? Are there nonpolar regions of this as well? You guys would be right if you speak up. Yeah, right? So we have these large groups that are filled with hydrocarbons. So I'll draw this on the wall again, but these were some examples of polar bonds, right? The end of this are this series of carbon-hydrogen bonds. These just don't, there's just not a particularly large difference in electronegativity between carbon and hydrogen, so therefore these bonds are not going to be polar. That means that these areas might be a little bit less likely to interact with a polar solvent, something like water. So water might be prone to interact here, but not here. So what may end up coming to matter is maybe the density of polar regions, the proportion of all bonds that are polar. We'll see that as we keep moving on. So again, we have all of these polar regions here, but we also have a fair amount of nonpolar ones as well. So we're going to go through this one then. Which of these things do you think is more polar? Right. Cool. So let's take a look at this. Most people are going with A. A couple people with B. I could see a valid reason why you would think that either of these could be correct. Anyone want to defend their choice? Yeah.
Ratios of polar bonds and non-polar bonds. And with water has two polar bonds and no non-polar bonds, so its ratio will probably be better than cellulose, which will evidently have a ratio.
Cool. All right. As we can, let me get rid of this thing. But that's absolutely spot on. It's this idea that it's not just, I know we see a lot of different polar groups here, but we also want to consider the fact that there are a lot of nonpolar areas of this as well. And polarity is not a simple, this thing is polar or this thing is not polar question frequently. You'll generally see me give some type C answers here, which are like, sort of. Sometimes those really are the right answer. Now, and what really comes to the forefront here is the question is which is more polar. And in this one it's going to be water just because the vast majority, or every bond in that water molecule is going to be polar, whereas maybe half, somewhere along that line, is polar here. Now that still makes this a pretty polar set of molecules in the grand scheme of things. So if we had a polar solute, so again, solute is something that's dissolved in a solvent. This thing here is one that you're going to be working with next week. This is chlorophyll A. Which of these two substances would it be more likely to bind or interact with? There we go. Cool. So again, we're a little bit split. This one, I will actually kind of select a right answer. I'd go with water on this one. The reason being that if this is your solvent and your water is very, very highly polar, it is going to outcompete the cellulose to bind to or to create hydrogen bonds or temporary interactions with the polar regions of this thing. Definitely kind of a foundational piece. I'd commit that one to memory here. This idea that water is probably the most polar solvent you'll ever see, which means that if you are looking at even a reasonably polar compound like this, it's going to outcompete something like cellulose in order to interact with that item, which is important when we start thinking about our choice of solvent, because water is not our only solvent here. And in this lab, we actually won't use it because it will make it very difficult to separate anything. Everything would just associate with water. So this here is hexane. We brought this one up already. What if we change the solvent to that? Which will the polar solute bind to more readily? There we go. So keep in mind this thing is polar. So we have a little bit of a split here, and I really want to make sure that we get this point. If you have a polar solute, it's going to interact with the most polar thing that you have available. On this, which of these items is going to be more polar, left or right? Polar, we're looking at. So that's going to be on the right here. Remember, our hexane is just a very long hydrocarbon. So your hexane is going to look something like this. There we go. Each of these is a carbon, 5, 6, and it has a series of hydrogens around it. And I'm not going to finish drawing the hydrogens on there, but I think you guys get the point. The idea is that there's not a single bond on here that we would count as even reasonably polar. The difference in electronegativity between carbon and hydrogen is a little bit less than 4. So in the grand scheme of things, it's barely polar at all. Whereas about half of our bonds in cellulose have a really strong dipole on them, making them polar. So actually, does that make sense why this thing is then going to be bonding there? Right, so the idea being that if we have something polar, it's going to be associating with a thing that is more polar. So let's then bring this back into our experiment. So if you guys were to run some paper chromatography, so we have our cellulose, that's this big piece of paper here, and we have hexane as our solvent, and we have the little green things, we can call that chlorophyll A. So this is a polar solute. What do you guys think is going to happen when we dip this thing in? I should also say that hexane is going to perform like water. This isn't a trick question. Hexane is going to travel up, be wicked up the paper. There we go. Okay. All right, let's take a look at that one. Awesome. So almost everybody said that the solute is going to travel with the hexane. Let's see. There we go. Okay. So, oh wait, so almost everybody picked B on that one, right? All right, so we have that. Let's actually go back a moment and talk about this before we run it. So there's this idea then, which of these items is our solute, our polar chlorophyll A here, going to associate more with? The hexane or the paper? Our paper, right? So what you just saw there, and this is going to be an observation, we saw that the hexane was able to travel up the paper, right? If I do this again. And there's probably a little bit of force right at the beginning where the chlorophyll A moves a little bit because the solvent is moving. But it's not going to go very far because it's going to bind pretty strongly to that paper and not really want to move at all. This is going to come up and be foundational. This is different than what we saw happen with the water when that was our solvent, because as the water was moving, these things were more attracted to the water. This is going to be at the heart of what we're doing next week in lab. Keep in mind, I'm also recording a video of this right now, so you will have access to this after class to go back through to help you guys out. So let's think about then, this is our other solvent we'll be using. This is that acetone. I'm not throwing something new at you guys. So what if we change the solvent to acetone? Which would our polar solute, this is the chlorophyll A right in the middle here, bind to? All right. So a little bit of a split here, but I wouldn't be surprised if some of this is just because we don't know which of these is relatively more polar. And that's fine. If you actually went down and counted every single polar bond, acetone would have a higher density of them than our paper. But one of the reasons we're using this one is there's not as large of a difference between these two as there was water. So we might actually be able to see a little bit of a difference. So as we go in and run this then, remember, the chlorophyll is going to be sticking with whatever the thing is that's more polar. What do you expect to happen when we dip our paper into our solution? Awesome. Awesome. So we'll stop that. And the vast majority of you guys should be here. And although I jumped the gun last time, this is actually what I was hoping for. So the idea then being that if the acetone is more polar than our stationary phase, I would expect that solute to move up with that, right? Cool. Good job predicting that, guys. Now, none of this, so that we've just sort of gone over some of the underlying principles of what we're doing, but none of this has really helped us separate two things out from each other yet. So we're going to kind of drive through that point now. So what will happen if we had a mixture of solvents? So we've got some of this stuff that's really nonpolar, some of these things that are pretty polar. And it will act as if it's one solution. It's not like this thing will separate out as we go. So what do you think? Which of these might it bind to more readily? Or neither? Okay. I'm actually going to bring the graph up. So we've got just about an even split here, right? Which kind of makes me think that maybe there's not a clear difference between the two. Right? Might it sometimes be attracted to the solution and sometimes be attracted to the paper? That's actually one of the places that we're going to be trying to get here this week, is we're going to be trying to find this point at which there's a bit of attraction between some of our solutes and the paper, and there's a bit of attraction between those solutes and the solution that we're working with, because then something cool will happen. So we'll stop this, X out of here, and move on to the next one. So what do you guys think would happen if we use that same mixture of solvent? So it's kind of attracted to the paper, it's kind of attracted to the solvent. Read this guy fully, and then answer this. Will the solvent remain in the dish? Is it going to travel at the same speed as the solvent as it goes up? Will neither of these things travel up the cellulose at all? Or will it travel up slowly? Okay, let's stop this one here. We do have a little bit of a split. Does anyone with either D or B who answered one of these two, do you guys want to defend your answer? Why do you think your outcome will be the one that happens?
Well, I chose D, and the reason why I didn't choose B was because I didn't think it was going to travel at the same speed because it's going to stop at different portions of the piece of paper based on like its polarity. So then I thought like D would just be the best like choice because it's going to move at its own pace. That's how you separate it.
Cool. So again, I think we're coming back to this same idea, and that's a really good way to voice this. If you have a mix of interactions with this, well, sometimes it's going to be moving with the solvent. Sometimes it won't be. So it'll probably move up this thing slowly. Let's take a look and see what happens. Cool. All right. So it's going to move at a relatively constant speed, just because sometimes it'll be interacting with the paper, sometimes it won't. Now, this is going to be really useful in your goal of separating out these pigments from each other, because what we now have is we have one that traveled partway up the paper, but not all of it. This becomes important when we add a second solute. So that top one, chlorophyll A, is one of the things you'll be extracting. It's pretty polar. That one on the bottom is carotene. Does that look polar to you guys? Raise a hands for yes. How about no? Right? Giant long hydrocarbon. There's going to be a bunch of carbon-carbon or carbon-hydrogen bonds. None of these things are polar. So that's not going to associate very much at all with the paper, right? Now, the trick here, and this is one of the harder conceptual points of this lab, this is going to start out in solution. It's more likely to stay in solution than it is to associate with some paper here. So when you run this thing, which one's the more polar solute going to stick to? So, or actually it looks like there's two different parts of this one. Two outcomes here. The more polar solute would associate more frequently with the cellulose, or the less polar solute would associate more frequently with the cellulose. Which of these things is going to stick more? So I'm glad I clarified that one anyways. Most people here are on a pick day. Again, we're coming to this idea that if you had two things in solution and they're running over a solid stationary phase, the polar thing is going to be more likely to interact with this occasionally. Not all the time. We're not saying that it's going to just interact with the paper and stay there. But if something's dissolved in our mobile phase, it's not going to come out of solution to bind to a very polar piece of paper. And that's going to be kind of the basis for what we're doing this week in lab. So if we have two different items, we have that green rectangle, which is representing cellulose. Which is representing chlorophyll A. That's our polar one. Our triangles are nonpolar. Which of these solutes do you think is going to be more polar when this happens? Right? So if you were just separating out two things, which of those is more polar? So I kind of ruined that question as I gave you guys the answer in part of my talking points. Most people, actually we ended up split 50-50. So we're asking more polar. So let's go back here. What do we know about something that interacts with our stationary phase here?
If it's interacting with it, it's going to be going polar.
Right. So our stationary phase is more polar than it is nonpolar, which means that if something is going to come out of solution and interact with it, that item is going to need to be polar. These triangles, they're not interacting with the stationary phase at all, right? Suggests that they're polar. That's not to say that they absolutely love our solvent here either. Our solvent does have some polar items in it. We're going to be trying to get a ratio of these things that will let everything stay in solution, but will also make our paper a little bit more attractive to our polar ones than our nonpolar ones, so that if you were actually running that whole paper chromatography, you would get something. Let's actually go back and rerun this. You would get a result like this, which you could actually use to separate these. Now again, these are, if we're zooming our scale back out and we think that this is like the size of a piece of paper and we got these things about 1 inch apart, you'd actually be able to cut this into 2 pieces and you've now separated chemicals based on their polarity. That's kind of the end goal of what we're going to be working on this week. Right. So, I think I saw a hand up. Did I miss a question here?
You answered it.
Oh, awesome. So, hopefully this has been a little bit informative. I am going to put this video up on Canvas, or a link to this video up on Canvas, so that you guys can come back and use this as you keep working on your pre-lab activity. It looks like we'll only get about 10 minutes or so for you guys to get started, but do get started with that and come back to this over the weekend as you have help. As you need help. Now, the modifications, you're only going to be completing part A of this. It's also not going to be handed in. The goal of this pre-lab is so that you guys can work through it and then use it on the pre-lab quiz, which will be challenging unless you're able to work through part A. You also have lots of help available to you. As a reminder, I'm sticking around both classes today, so today for about probably a half hour after class, and then I have an evening class that ends at 4:40. I was going to stick around for at least 20 or 30 minutes then as well. You guys are, sorry, it ends at 4:25, so I'll be here from about 4:25 to 5 o'clock or so. You guys are more than welcome to come by. I'll be meeting right across the hall or in the classroom, depending on whether someone's coming right in after us. Questions before we break this back off to everybody? No? All right, start working in your post lab and call me over if you need help.